Stanford University.
Welcome to the third mini med school session. It's great to see you all back. I'm Sherry Wren, who co-directs the course with Phil Pizzo. Gil Chu's slides from last week are now up on the website, so you're welcome to go and look at them. It gives me great pleasure tonight to introduce Jill Helms, who's going to talk to us about stem cells and tissue regeneration, which I think is something you can't turn on the TV or the radio without hearing those words for the past, 8 years. And Jill has sort of an interesting background for a medical school because she actually is not only a PhD that she got from the University of Connecticut, but she's also a dentist. And she got that degree from the University of Minnesota. And Stanford does not have a dental school. So she has unique talents, though, and we were really happy to recruit her away from UCSF, where she served on the faculty for 8 years. She's currently an associate professor of surgery here, and she's very interested in craniofacial development and also regenerative medicine. She does really exciting research, and if you read her CV, my favorite thing she does is she works on a set of protein called the sonic hedgehog proteins. Now, I think that anybody who can work on something called sonic hedgehog, I'd love to know how that protein got named, because somebody was definitely having some fun with that. Today, what she's going to do is really introduce you to the terminology used in stem cell research, and then share with you some of the recent advances that have been made, and we'll discuss how stem cells could really be used for medical treatments and this whole new field that is now being called regenerative medicine. So I'd like to welcome Dr. Jill Helms.
Thank you. Thank you so much. What a pleasure to be here. I see some familiar faces, a lot of new faces. I have gotten the best subject to talk about in this whole mini-series. I must say, to be honored with the topic of stem cells and regenerative medicine is like being handed the best topic. So I hope to excite you with the same enthusiasm I have for this subject. So I'm going to start with a couple of questions. First of all, by a show of hands, how many people know someone who saved umbilical cord blood after pregnancies? Raise your hands high. Okay, I'd say about a third of you, at least a third, maybe a half, okay? How many people know someone or themselves have a condition that they believe could be treated by stem cells? Yeah, that's impressive. Yes, Nancy Pelosi said every family knows that they're one phone call or one diagnosis away from needing what stem cell research can yield for us. I think that's so true as we go along these days. Last of all, how many people in here voted for Prop 71? You remember what Prop 71 was? How many people voted in 2004? Well, over 7 million Californians said yes to Prop 71, which funded stem cell research in this state. And I want to tell you a little bit about what we're doing, not only in stem cell biology around the world, but also what's happening here in California, and most specifically here at Stanford, where we lead the institutions in California in stem cell funding. So in New York Times, I had a friend of mine look through the last 3 years and found out how many articles mentioned stem cells. 604 in just the last 3 years. And the amazing thing is the distribution of these articles were in everything from science to business to politics. My favorite is 11 mentions of stem cells in the style section, which I read religiously. So 12 are in the obits. Anyway, hopefully not. So you can see that stem cells are big news. There's no doubt about it. And the field of regenerative medicine is built around stem cells. And I'll tell you a little bit about how those two topics sort of segregate. But tonight I want to tell you about what stem cells are, what makes them so unique, and most importantly, do they really have the power to transform the way we practice medicine today? And I'm going to hopefully convince you that, in fact, we absolutely do. So first of all, I'm going to tell you how the talk is going to be broken out. So if there are those among you who decide one part of the talk is a snoozer, you can up and leave and come back for a later part. Hopefully you'll want to stay. First of all, I'm going to tell you why we need regenerative medicine. Then I'm going to introduce you to the lexicon of stem cell biology, make you familiar with some of the key, maybe the key nomenclature that accompanies stem cell research. Then I'm going to talk about the concept of regeneration. Why is regeneration different from repair? And what can we learn from animals that do regenerate very well? Then I'm going to go on to talk about stem cells in particular, cloning, which is the basis for stem cells, and some real-life applications that we think are just around the corner. And then last, I'm going to tell you about Stanford and stem cells, one of my favorite topics. So first of all, why do we need regenerative medicine? This was an article from the Washington Post a couple years ago, and they heralded all of the advances that have been made in the machinery to replace missing or damaged parts for the human. Here you see a cochlear implant. Here's a couple of others they mentioned: dental implants, corneal implants, hip implants, artificial hearts. With all this technological advancement, why do we need regenerative medicine? And I think that I'm going to start with the concept that as good as these machines are, there are limitations to all of them. How good is good enough? Does this young man who came back from Iraq and needed an amputation, is this good enough? He can walk. But there are limitations that go far beyond what we think about aesthetic reconstructions. For example, I want to emphasize that hardware is not equivalent to software. This is a very famous athlete. Does anybody know who this is? Oops. Barbaro, that's right. That's his leg. And you can imagine the best veterinarians in the world were working on his leg. And you can see the amazing amount of hardware that has been put in there to hold those bones together. Regardless of the strength of that implant, that bone plate and those bone screws, I can tell you that that material is not as strong as bone. And what you see on the right-hand side is an illustration of strain fields within materials. And in this case, it's strain fields within bone. And the important thing to notice is that bone has the ability to adapt to loads and can bend and conform to a load that's excessive. These plates have a failure point. I think any engineers in the group will understand what I'm talking about. And they oftentimes do fail. This one failed as well. And of course, we know what happened to poor Barbaro. There's another reason why man-made materials aren't nearly as good as our own tissues, and that is adaptability. I show here just two examples of the ability of bone to both resorb, that is, be taken up, and be reformed. This process is going on all the time in your bones, in your body. So every 7 years, the skeleton you have now will be completely replaced with a new skeleton. And the advantage of that constant renewal is that if you have excessive loads, the bones will adapt to that load. If you have less loading, we know that you can lose bone mass. But this process means that defects in the bone structure will eventually be taken out by this remodeling process. This, of course, is not something that a man-made material will do. By the way, interrupt me if you have any questions. There's another important difference between man-made materials and our own tissues, and that is the ability of a man-made material to integrate. Here you see an example of ligaments, muscles, and tendons that all make bones functional. So we can't just replace the structure of a bone with a steel rod, no matter how strong it is, because we'll never get the functional integration of that steel rod with muscles and ligaments and tendons attaching to it. So all of these things tell you why man-made materials have limitations and why those won't nearly be as good as our own natural tissues and organs. So then you might ask, well, what about transplantation? Because there it is, we're not using an artificial heart, we're transplanting one heart from a donor into a recipient. And you're right, those tissues have much more adaptability. But we all know the biggest disadvantages of these tissue and organ transplants. Anybody who's on a list will tell you that the limited number of organs and tissues for donation mean that a lot of people die needlessly. there's another issue, and a lot of people don't really keep this in mind, and that is if you receive an organ, it's from another person, and that means that your cells can recognize the cells that are comprising that other tissue as foreign. And that means if you didn't give a patient immunosuppressive drugs, they'd eventually reject that tissue or organ. And so these immunosuppressive drugs are something that patients have to be on for the rest of their lives. And there's a very high cancer risk associated with drugs like this. Of course, if we're talking about somebody's heart, you can't live without a heart, so you'll run the risk of taking the drugs and having cancer later on. But you can understand now why other kinds of tissues that aren't going to be life-threatening are rarely transplanted. Things like people, maybe you've heard about the transplanted hand or the transplanted face. Have you heard about those things? So those are things you can live without, right? But still, some people opt to take that risk because they're so aware of the deformity or the limitation of life without that organ or tissue. So I hope that I've made it clear that regenerative medicine will address these kinds of limitations, but there's one other thing. Besides being able to replace tissues or organs for which we have man-made materials or maybe the ability to transplant materials, regenerative medicine also seeks to address diseases for which we know no cure. And that is an amazing hope that we hold for the future. And I really do believe that it's very close at hand. So now I'm going to move on to the second part of the talk. And if you guys don't interrupt, we're going to be out of here by 8 o'clock. And that is the concept of regeneration. And this, what? Oh, I'm sorry. Oh, yes.
If you said that you basically you were on a lifetime those drugs. What if could a twin donate something and then would the recipient accept that more readily?
Right. So the question is, can a twin be a donor? And will the recipient accept those tissues more readily? Yes. Yes, that's absolutely true.
Have to take the drugs?
That's a good question. Would you still have to take the drugs?
So it depends on whether you're an identical twin or a fraternal twin.
Yeah.
If you're an identical twin, you're biologically the same. And actually, the first kidney transplant that was done in the United States was done between identical twins. Then you would not need to take immunosuppression. If it's a fraternal twin, you do have differences that the body can recognize, and then yes, you would have to take immunosuppression.
So it's about being genetically identical. Okay, yes?
I was kind of fascinated when you said that every 7 years your skeletal system is basically renewed.
Mm-hmm.
How then can you see in an X-ray a fracture that happened many, many years ago?
Right, that's a very good question. Why does a fracture that happened many years ago not get completely remodeled? Well, that efficiency at remodeling your skeleton decreases as you age. And so in areas that have fractures that never heal, the problem there is that the mechanism for repair and then remodeling is disrupted. And so typically those kinds of fractures that don't heal do so because they lack a blood supply. And if you have that kind of problem, then that area of your skeleton will not remodel. And of course, with osteoporosis, we know that the bone resorbing activity is greater than the bone forming activity, and that's why you start losing bone mass with the disease osteoporosis. Okay, yeah?
Is the same thing true with scar tissue?
Yeah, is the same thing true with scar tissue? Does it repair or remodel like that? Unfortunately, no. And that's a big problem because scar tissue, and I'll talk about this in a little while, scar tissue has very different properties than the initial tissue that formed. And a lot of tissues don't remodel quite like bone does, but certainly scars do not. And so I'll bet anybody can look and say, I got that scar when I was 7 years old. It'll stay there for the rest of your life, or the place where you got your vaccination. Those scars never remodel, and that means that when we have tissues that heal through scarring, they'll be there forever. Okay? So now I'm going to, oh, I'm sorry, yeah?
So why do they tell you you should do weight-bearing exercise to help prevent osteoporosis?
Right.
How does that affect that mechanism?
Right, so why does weight bearing appear to help with osteoporosis. So weight bearing is one way your skeleton senses that it needs to build more bone. So like a tennis player who always uses his, like Roger Federer, not me. So when he uses his right hand, his skeleton of his serving arm is actually larger. So actually mechanical force like walking or weight-bearing exercises or something like tennis will make a skeleton grow. It will make that part of your skeleton grow. And so that's in an effort to stimulate the bone-forming capacity of the body versus non-weight-bearing activities.
Question?
Yep.
I see nowadays a lot of people doing knee replacements and surgeries. What is your opinion on that?
Right, so what's my opinion on knee replacement, knee surgeries? This is from not a surgeon's point of view, is that wait as long as you possibly can before you do it. Because unfortunately, these implants tend to loosen over time. That seems to be the case in almost all kinds of implants like that. And so that means when you have the surgery, you have about 15 years, would you say, Sherry, is it about 15 years, before you need to have another one? And the problem is every time you put in that implant, you have to take a little bit more bone away. And as that goes along, of course, you're going to eventually have not enough bone to hold an implant in place. So these kinds of man-made replacements, I mean, sometimes pain is the distinguishing feature which says now it's time. But a lot of people try to put it off as long as possible. Okay, so now I'm going to move on to the idea of regeneration. And we're very lucky to think about regeneration because nature does it nearly flawlessly in some organisms. So first I'm going to talk to you about two features that stem cells have that make them unique. So here's an illustration of a stem cell. The first unique feature of a stem cell is that the stem cell can become any cell in the body. And I've illustrated that here with a stem cell with a little halo around it. And I've showed you the kinds of tissue that we typically think about a cell being able to make. That is muscle, bone, fat, and cartilage. 3 of those we want, one of them not so much. But you can imagine that the ability of a stem cell to differentiate into any of those cell types is really quite remarkable. Now, I want to tell you how remarkable that is by telling you that when a bone grows, it's a bone cell giving rise to another bone cell. That happens because the cell divides. This is a stem cell that can become any one of those kinds of tissues, you see? So that makes it very unique. Now I want to give you an example of that from science, and this was published just a couple of years ago, and it uses the tissue of the breast. Now here's a schematic of the breast, and this has now been shown in a number of tissues, but the breast is a good example. And in the breast, you know that its primary function is for lactation, and you can see the ductal structure in a mouse breast here on the right-hand side. And that entire very complicated lattice work is comprised of many, many different cell types, and I'll show you just 3 of them. There are alveolar cells, and those are illustrated here as lining a lumen or a tube. There are the ductal cells that lie on the outside of that lumen that serve as sort of the support. Then there's muscle that surrounds the ductal cells, and then there are the milk-producing cells in the middle. There's one other cell type that's found in the breast besides these, and that is the mammary stem cell. That is a stem cell that has the ability to give rise to all of the cell types I just told you about. And here's the proof of that. So I'm not going to tell you how they isolated the mammary stem cell, but I'll tell you that they did it from a mouse that was genetically manipulated in a way so all of the cells from that mouse would turn blue if they're exposed to a particular chemical. So all of the cells from this mouse are labeled so they're blue. And these researchers isolated the cell that they call the mammary stem cell, and then they put it back into a recipient. Now, it turns out the recipient was not genetically marked, so it was blue. It had normal colored cells. And then they looked at the developing mammary gland. And remember now, maybe you don't know this, but the mammary gland grows during pregnancy and during lactation, and then it recedes and then it grows again. So in a mouse, they made this mouse a mouse that was pregnant, so the breast was then growing. They put the cell back in. And when they transplanted a single cell, which they call a stem cell, into the recipient, here's what they found. All of the cells in the breast were blue. The ductal cells, the milk-producing cells, the alveolar cells, all of the cells were derived from that one cell that was put in. And that's proof of the concept that a stem cell can give rise to any cell type. We call that differentiating into any cell type. And this ability is called pluripotency. It's a very unique feature that's reserved for stem cells, whether they come from embryos or adults. Yep?
Does that mean there was only one mammary stem cell operating?
That's a good question. Does that mean that there's only one mammary stem cell? No. There are multiple mammary stem cells, and they tend to be activated by injury. It's just that in this experiment, they wanted to prove that a single cell could do it. So that's why rather than take the multiple mammary stem cells that they can find, they took a single cell only. Does that make sense?
But I would think the mouse would have its own mammary stem cells that were not coated blue.
Very good.
Why?
Yes.
The blue one was obviously injured, so maybe
That's exactly right. So that's a very good question. So the host animal has its own mammary stem cells. Why didn't they contribute? Well, in fact, you do have to have the injury and the removal of part of the breast tissue in order to get the new cell to integrate. So if you just inserted the one blue cell in there and didn't disturb the recipient's stem cells, then you get a patchwork. It's called a chimeric mouse. But in this case, they removed a part of the breast, and so in doing so, they removed the recipient's own stem cells. A very good question. If you want a job in the lab, come see me afterwards. All right, there's another unique feature of stem cells, and that is that they can duplicate themselves. Now, that is a very unique feature, and I've illustrated it here, but sometimes people think that what this means is, like I was saying, when the bone is injured, one bone cell gives rise to another bone cell. That's not the same. This is like if you have children, you might think, well, that's like one cell giving rise to two cells, a parent giving rise to two kids. That's not what this is. This is you giving rise to you again, exactly you again. And that unique feature is called self-renewal. And it seems to be limited to stem cells. And now I'm going to give you an example of that ability to self-renew. And I think most people have probably heard about bone marrow transplants. This is clearly a way that stem cell research and regenerative medicine have already been established in the clinic. So here you see the inside of a marrow cavity. And what I hope you appreciate is its tissue. There's not just, it's not like your bones are empty and it's a pipe full of blood. This is actually cells and tissue. And in a bone marrow transplant, this tissue is harvested from a donor, and here you see that happening right now. And then it's injected into a recipient. And like in the case of the mouse, the recipient is usually very ill, and their own bone marrow has to be removed in order to allow the new bone marrow to integrate. And here's the reason why that's possible, why it works, is because in this bone marrow are contained all of the cells, the stem cells that give rise to all of these different kinds of blood cells, as well as some, I don't think you can see it here, as well as the precursors of bone. So when the doctor takes the bone marrow from a donor and puts it into a host, what they're doing is transplanting stem cells. Now we know that both the donor and the host can survive this. So the donor can do just fine because their stem cells can renew. These hematopoietic stem cells and their ability to self-renew was one of the first kinds of stem cell self-renewal that we really understood. And I think it's beautifully illustrated in this model of bone marrow transplantation. So these are the two unique features again. The first is that stem cells have the ability to self-renew And that capacity appears to be limited to stem cells. And the second feature that distinguishes them from all other cells is that they're pluripotent. That is, they can give rise to any cell type. Yeah?
So does that mean when they go through mitosis, they don't make mistakes?
So you asked whether when they go through mitosis, they don't make mistakes. Well, of course, any cell can make a mistake. But
supposedly they live forever, these cell stem line.
some stem cells are very long-lived. Other stem cells are very short-lived. So we used to think the length of a cell's life was an indicator of whether it was a stem cell or not. Now we know that that's not true. Stem cells can make mistakes, and we think that cancer is an example of a stem cell making a mistake. Because, well, it may be a different kind of stem cell, but it appears to be in a tissue, so we call them tissue-specific stem cells. And they're basically stem cells that keep on hearing the signal self-renew, 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 and they never hear the signal differentiate. And that's what makes it a cancer stem cell. We think that they're a unique kind of stem cell with a defect. Yes?
You said the stem cell can self-renew. Now, other cells divide.
That's right.
Each cell is identical, so what's the difference here?
Yeah, it's a difficult concept, but it's sort of the parent giving rise to two kids. That's what tissues do when they remodel. They give rise to two cells that are already differentiated. They're already bone cells. Whereas a stem cell doesn't give rise to a cell that's different from it. It gives rise to a cell that's exactly the same. Does that, I'm not sure if I'm answering. I can get into more detail.
Think of any cell in the body, though. I mean, you take an individual cell through mitosis, there's then two cells that are both
Then you have
identical.
two cells, and they're both differentiated already. Like a heart cell or a muscle cell giving rise, dividing into two muscle cells. A stem cell, when it goes through that process, it still gives rise to a cell that can give rise to any other cell type. Whereas a myoblast can only give rise to another myoblast. Yes?
In bone marrow transplant replacement, do you need immunosuppressant drugs?
Well, oftentimes not, because a patient, the recipient, has had a radiation to remove all of their endogenous or their own bone marrow. Usually they're very ill as a consequence of this.
How much bone marrow do you remove from the donor?
How many cc's? Sherry? Oh, she winced.
I've done 1 million bone marrow transplants in mice. I can tell you that. No
Yeah, we can tell you everything in a mouse. But
yeah,
what would
anything
you
in a mouse,
guess?
10
In a
to the
human?
6th in a mouse.
Okay.
It's
How many
probably not.
cc's?
It's not that much. I don't know. I'm sorry. It's a fairly small volume. Yeah.
I bet it's under 100 mils for sure. I'm
Yeah.
guessing somewhere around 50.
Yeah. Sorry.
A
If you were
small
a mouse, I
cupful.
could tell you.
Yeah. So we know everything about mice. Yeah.
Is the self-renewal also called mitosis with a stem cell versus the differentiated cells?
So is it,
Process the same?
is it called mitosis? Yes, I believe it's still called mitosis, although people usually differentiate it by saying self-renewal. But I believe you can still call it mitosis, yeah. Yes?
With the bone marrow transplants, don't they often take it from the person that has the cancer
So you're asking about the,
and then put it back into them?
so you're asking about the bone marrow transplant, where do they get the donation of the new bone marrow? No, it's never taken from the diseased individual, because that bone marrow is essentially diseased. And so, I'm sorry?
So there were autologous bone marrow transplants. That's what that's called.
Yeah, but
it was the first place you heard about it was actually in breast cancer. There was a huge thing, actually court cases, because insurance companies denied payment for autologous bone marrow transplant to treat breast cancer. It then was shown in studies to actually not be of benefit. So to my knowledge right now, there may be some conditions where autologous may be sufficient depending on what you're trying to treat, but most of the bone marrow transplants for things like leukemia or other diseases of the blood are coming from other donors, if that answers your question.
And again, with the issue of compatibility, they try to typically use people who are closely related because the cells, as they get introduced into the patient, still have markers on them that distinguish them from the donor or from the host. Yes?
Well, just speaking about the different kind of transplant I actually work with, we do a significant number of autologous transplant, and it depends of what is the problem that brought you to transplant. You can have a stem cell transplant, not because you have to have a different immune system, but because you might have to receive otherwise killing dose of chemotherapy, and you just need a rescue from that, what becomes a cure dose that would kill you by immunosuppressant.
That's a very good point.
It's sort of a rescue transplant.
Yeah. It's called a rescue transplant.
Autologous transplant, neuroblastomas, you do that. A significant number of it, I would say it's about 40% of, at least in the pediatric population, we do autologous transplants.
Come on down here. That's a beautiful answer, and that's from real life. Anybody else want to? Yes?
Sorry, I'm still confused between
She can help you.
I'm still confused between mitosis and self-renewal at the replication level. Last week we heard Jeffrey Chu talk about DNA replication and a good discussion of 3' and 5' homologous replication. What's different?
So what's different about this process? Well, I can tell you that the short answer is we're not entirely sure. But the longer answer, and a little bit longer, is that stem cells, unlike other cells, seem to have to actively repress differentiation. When they divide, when cells typically divide or undergo mitosis and give rise to other cells, they already are expressing genes that have to do with their differentiation. And stem cells have to actively repress this. Now, we used to, I'm sorry, go ahead.
Sorry, you're saying expressing genes. All that's going on is a simple chemical process of replication. It's not expressing the gene, it's replacing or duplicating. So there's no protein being formed. That's what's confusing me. It's just a simple chemical process.
I guess what I'm trying to emphasize here, and I might be missing your point, but what I'm trying to emphasize is the difference between self-renewal and differentiation, not what is mitosis. So what I'm trying to emphasize is that unlike differentiated cells, when they divide, one thing that I think that the mitosis part or the replication is indistinguishable as far as we know, but there's a caveat there. But what makes these cells unique is not that they undergo mitosis, that's true of all cells, but rather that when they do so, their progeny is at least one of them, one of the two progeny is a stem cell again. And there has to be a mechanism in place for making that happen that's very unique. We can talk more later, I'm sorry.
If you are a skin cell and you divide, you become a skin cell again. You don't become a bone cell or a blood cell. Whereas this cell keeps that ability, which is a miraculous ability, to become any cell. So it is all the mitosis, but a skin becomes a new skin.
And a stem cell becomes a stem cell. But I don't see what's different. They're both doing what they naturally do, is replicate. What's different about this replication?
I hope to have convinced you at the end of this of what's so special. Yeah?
Do we know how we initially get our first stem cells?
Oh, that's a very good question. Yes, we do know something about how we get our first stem cells. And I'll show you that as soon as we finish the mitosis part of this talk. I am going to answer your question sometime, yes?
So can you describe what's the process when a stem cell decides suddenly to be a muscle cell? Or what's that what initiates the differentiation? And after it differentiates, does it then, after it differentiates into a bone cell or whatever, does it then make stem cells when it undergoes mitosis or other bone cells?
Right. Okay, so two questions. How does a stem cell know what to do, or how does it get its instructions to, okay, now you differentiate into bone? And then once that cell is differentiated, can it go, oh, I'm going to go backwards. Okay.
Or make baby stem cells.
Right, or make more stem cells. Okay, so the first question is very complex. We know something about how cells choose to become differentiated, but it involves the turning on and off of multiple genes, gene regulatory networks that lead those cells to differentiate. And I can give you names of genes, but what exactly triggers that differentiation is not always clear. When we transplant stem cells from one tissue into another, we have some ideas of what those signals are, because they're those signals that come on very soon after the cell has been transplanted. Sometimes we can even drive those stem cells to differentiate by adding certain growth factors. But it's this complex milieu of the environment in which a cell finds itself, growth signals and differentiation signals that the cells are getting that lead them to differentiate. So we know something about how they differentiate. Now, once they differentiate, 2 years ago, I would have told you, no, they're done. They're differentiated, that's it. In the last year and a half, we now know that's not it. They can go backwards. It's phenomenal. It's truly phenomenal. I'll tell you about an example of that towards in the second part of the talk. Yeah?
You mentioned about the mouse, you called it a memory stem cell.
Yes, memory stem cell.
Does that refer to the location where it's found, or has this cell already kind of differentiated into becoming any type of rest cell that it might
Yeah,
be?
very good question. So the word that describes the stem cell, like mammary stem cell, indicates the tissue of its origin. So that means that within the breast, they found the stem cell. There's epidermal stem cells found in the skin, there's kidney stem cells, etc.
So if I change the environment and put that cell into a bone state, it could become a bone cell.
Right, so the question is now, if you take a mammary stem cell, can you put it anywhere and it'll become a bone cell, a muscle cell? We used to think absolutely. And now, just in July, was published a paper that suggests that rather these stem cells in adult tissues, like this is a mammary stem cell, so it's tissue specific, that they seem to remember where they came from. They remember even if you put them in a dish and you passage them over and over and over again. You'd think a cell would not know this, but they do seem to remember, and it's phenomenal that they have this memory. This memory, we think, is encoded by a set of genes called Hox genes. And that seems to imbue stem cells with some kind of memory of where they came from. It doesn't mean that they can't make other kinds of cells, but they tend not to do so normally. So if a cell came from a certain germ layer called the ectoderm, it's most likely to give rise to tissues or cells in tissues also derived from ectoderm, like skin or bone. Whereas cells derived from mesoderm tend to give rise to mesodermally derived tissues. This is a restriction that we were unaware of before. And I'll show you an example of that in a little bit. Okay, now somebody asked over here, I think, where do you get stem cells from? That's a very good question, because of course if you want to study them, you must have a source of them. And I'll bet a lot of you know that the previous administration had something against using embryos for stem cells. And now I think this happens to be a mouse embryo, but it looks very much like a human embryo. And you might look at this and say, okay, so that's why people are upset, because this is where you get embryonic stem cells. You get them from an embryo. But what I want to emphasize here is this is not the stage at which you get stem cells. This looks like a baby mouse, well, to me. This is where you get stem cells from. It's called a morula. That's from the Latin meaning mulberry. It does look like a mulberry. This is before implantation. This is the source of embryonic stem cells at this stage. And let me show you a little movie now that shows
With
in
exquisite
some
grace,
detail
one cell becomes
how you
two.
get a morula.
Two become four. Each duplicates the original, unique DNA. The enchanted progression of cell division continues. For 5 days, the embryos are monitored. Finally, the division creates masses of cells known as blastocysts.
Okay, 5 days. 5 days in culture, and you get this, the blastocyst. It comes from the Greek word meaning bud. And as you can see, there's a small group of cells right here called the inner cell mass, and that's schematized here. This ball of cells right here, called the inner cell mass, in that little thing called a morula, that is where the stem cells reside. And that inner cell mass contains all of the embryonic stem cells that will give rise to every tissue in the body. Now, there's something very unique about embryonic stem cells. They're not found within tissues, as you can see. They're at the beginning, before there is any tissue. And the thing about embryonic stem cells, which really distinguishes them from adult stem cells, are, first of all, adult stem cells have tissue-specific properties, mammary stem cell, bone stem cell. But the greatest potential is found in embryonic stem cells that have no tissue specificity. And here you can see that some kinds of, or adult stem cells can form some kinds of tissues. We call them multipotent, whereas embryonic stem cells are pluripotent or totipotent. This is what makes embryonic stem cells so valuable, because they have the capacity to give rise to all tissues, all cell types. Now, something that I want to point out right now is, how do you ever get adult or tissue-specific stem cells? We don't know the answer, but what it appears to be is during development of an embryo, cells are somehow set aside in every tissue where they stay in a quiescent state until disease or injury seems to activate them. We don't know how they get there. We don't really know where they reside. We have to search for them using very technologically advanced methods. But they seem to reside in every tissue, meaning that every tissue has the capacity to regenerate at least a little.
At least what?
At least a little. There's oftentimes other processes that go much, much faster, and that's why we get scar tissue. And I'll talk about that in a few minutes. Yeah?
Does that include the heart?
Yes, that does include the heart. They now have identified cardiac stem cells. Yep. And in the brain, where you might have thought that that was it. You know how you're told, don't drink because you're killing your brain cells, that's all you got? Turns out, few more. Okay, there's another big difference, and this is important when you're doing research, and that is that adult stem cells, when you grow them in culture, have a limited lifespan, whereas embryonic stem cells can grow continuously. This becomes very important if you're trying to study them. And the last thing is that in adult tissues, the number of stem cells is very limited, whereas in embryos are abundant. So here's the news that maybe it's bad news. As we age, stem cells stop being stem cells. They tend to differentiate until the proportion of stem cells is significantly reduced, and they make up a very small percentage of our total cells. And I say that with resignation as I start to look more and more like that woman with the bathing cap. Okay, now what I just told you is true in humans. In some animals, they retain the ability to regenerate throughout their life. It is a remarkable ability. And now I'm going to talk about those animals. Oh, yeah?
So the Bush administration,
yes,
I think limited it. It wasn't that there was no stem cell research, there was a certain number of existing lines.
That's right.
Why does the world need more than even one line? I mean, if they regenerate forever, why isn't that good enough?
Right. So so it's a fair question. There were a few lines. Most of them turned out not to be so good. But why do you need more than one? If you can make, it's a goose that lays the golden egg, you can keep on getting more. The problem is in the actual culturing of these cells. And now I've got to explain a little technical detail. When you take that inner cell mass and you replicate it and keep it growing, you have to give it the growth factors that it needs to survive. And the way that's done is by putting those inner cell mass cells on top of something called a feeder layer. Those feeder cells are not supposed to contaminate the inner cell mass cells. They're supposed to stay separate. And the feeder layers are basically supporting the stem cells, providing growth factors. But it turns out in almost all of the cell lines, those feeder layers eventually contaminate the stem cells. So the lines that were produced many, many years ago tended to have a lot of flaws in them. And so they weren't very robust as embryonic stem cells. Now, there were certainly places where people could generate new cell lines, and that has been done, and with a lot more care in keeping these feeder layers separate. But those were done with institutional money that was not federally supported. So, for example, Howard Hughes Medical Institute supported a lot of that the development of additional stem cell lines. So that's the main reason. Also, some of those stem cell lines were from adult stem cells, and I've already told you why those would be limited in their potential.
Yeah? They're taking skin cells and then taking them back to an embryonic state to then fast forward them to other cells. Is that happening?
Yes, that is happening. That is called reprogramming, where you take a differentiated cell and make it look like a stem cell. And I'll talk about that in more detail, but how was that achieved? But at the time, I would say this was about 2004, that was not even on the horizon. Okay, so regeneration. Some animals have enormous capacity. We do not. Why is that? Well, one way we can learn about how we heal tissues versus how these animals heal tissues is to study these animals. And there's a bunch of them that are really good at regenerating. And this is one in particular. This is a newt or an axolotl. They come by very many different names. And I want to show you a video that was taken from the Howard Hughes Medical Institute, and where they videotaped this process of regeneration. Now, I'm going to turn off the sound on this simply because the person narrating talks on for quite a while. Here you see the axolotl has had the limb cut off, and now you watch it over the course of 60 days completely reform. Now, this is schematized here. You can see now the axolotl can move its limb. It's showing you the cartilage, the muscle, the nerves that make it movable. And then the experimenter comes along, whoop, snip, okay? And now what you see here is the formation of something called a blastema, a wound blastema that forms at the tip of the amputated limb. And what they're showing you here now, this streaming, are cells, stem cells, coming from the cartilage, the muscle, and the surrounding soft tissues and the nerves that flood into that blastema, and then through mechanisms that we're not completely fully aware of, they reorganize themselves and regenerate a complete functioning limb. As many times as you can stomach cutting off that limb, it will regenerate. I know. That's, ooh. Okay. So where does the new limb come from? It comes from the blastema. Here's a picture of that little blastema, and there's a tissue section through the blastema. This comes from the Greek word meaning to sprout. The entire ability to regenerate all the tissues of the limb are contained within that little group of cells. So here's an experiment that was just published in July 2009 that helps us understand how is this possible. So here you see I've schematized that limb again. There's the cartilage, the muscle, and the nerves that comprise the limb. And there are three populations, I've showed you three populations of stem cells, one from each of those tissues: muscle stem cells, cartilage stem cells, nerve stem cells. And when the limb is amputated, what we now know is that each of these tissue compartments contributes stem cells to the regeneration of the limb. And each of them keeps track of where they came from. We used to think that one cell could give rise to all of them. Now we know that each tissue delivers its stem cells to the blastema, and then through processes we're not completely aware of yet, they reorganize themselves and generate a functioning limb. By understanding in a salamander that there are tissue-specific stem cells, we learn something very important. We learn that each tissue contains stem cells in salamanders and in us. That's the amazing thing. The process is conserved all the way from salamanders to humans. The difference is that they can reorganize these stem cells, and most importantly, there appears to be an unlimited number of stem cells. Like I said, you could cut that limb off again and again and again. We seem to have a limited capacity to produce more stem cells. What if we could jumpstart the stem cells that we do have within our tissues and maybe make a bigger pool of stem cells? That is feasible. Yeah?
So are there any long-lived animals that have this kind of regeneration capacity? Because it seems like if you're living longer, you accumulate more DNA damage, and then having this kind of capacity is a recipe for horrible kinds of cancer.
Right. So the question is, if long-lived animals maybe don't have this regenerative capacity, so the ability to regenerate would be associated with a short-lived animal because then you couldn't accumulate the mutations. Right. So we used to think that that was it. That it was just that simple, that animals that live for very short periods of time have a lot of regenerative capacity, but we've somehow winnowed that out because we live longer and then we could have more mutations. It's not that it isn't true. It is true that when you live longer, you accumulate mutations, and we heard last week about that. But it doesn't seem to be the case that long-lived animals have fewer stem cells than short-lived animals. For example, squirrels. Do you have any idea how long a squirrel lives? It's like something like 4 years. It's long, maybe even longer than that. I mean, it's long to me. Rats, which look just like squirrels, except for they don't have the furry tail, they're much shorter lived. And yet they both seem to have the same number of stem cells. So we don't know why that is. It's maybe just an association, but not causal. Okay, so now I remember a question about, well, what's the difference between repair and regeneration? So we don't regenerate after a lot of wounding, after some kinds of wounding. Some tissues do have an ability to regenerate, but I'm showing you one that does not, and that is skin. Despite the fact that our skin turns over constantly, when we suffer a deep injury into the deeper layers, not the superficial layers, but the deeper layers, we form scar tissue. And at first you might think, well, that's okay, scar tissue is not so bad. Well, there's a fundamental difference between these two, and I'll tell you why, and then I'll show you why. When we repair something, we replace the disease or damaged tissue with more tissue, but it doesn't necessarily have the same capacity as the original tissue. Regeneration is different. This means to reform in whole or in part the original tissue. So here's what scar tissue looks like. Scar tissue is not like normal tissue. As you can see from this soldier who was burned in Iraq, this scar tissue has different functionality. It lacks sweat glands, it lacks hair, it lacks elasticity, and as a consequence, it is not a functional replacement. Now, besides the aesthetics here, you have to understand that this scar tissue, the ability to form a scar tissue versus regenerated tissue, may have evolved because we had to develop a way to quickly close off wounds. But these are conjectures. We have no proof that that's why we generate scar and salamanders regenerate limbs. But I can tell you that there are a lot of tissues where scar formation is devastating. Think about spinal cord injuries. There's a scar that forms at the site of injury, and neurons can no longer pass that scar. And as a consequence, we have paraplegia. After stroke, we develop a glial scar in the brain, and functionality is reduced in that area. And we know after heart attacks, we form scar as well. And you might think, well, you know, that's better than having a hole there. But you have to realize that that scar tissue now compromises the ability of the heart to function. All of these kinds of healing, this mechanism through which scar tissue forms, are substandard and obviously not regeneration. Yes?
In case of salamander, the ability to regenerate, is it limited to their limbs or it extends to all of their body parts and tissues?
They have, so you were asking a question about, is the salamander limb the only part that regenerates? No, it's not. They regenerate their tails. If you cut the optic stalk, they'll regenerate that so they regain sight. There's multiple parts that you can cut off and replace. It's just that the limb is used, first of all, because it's relatively easy to get at versus the optic nerve or something like that. And second, because so many tissues have to contribute and organize themselves properly, and then you have a real readout, you know, it moves, of whether you've got full regeneration. That's why it's usually the limb that they use.
Right, but their internal body parts, like they must be, after their life, they must be dying, right? Because certain body parts are not functioning well, so do they also regenerate?
Yes, internal organs can regenerate as well. In zebrafish, for instance, you can cut, I think it's 2/3 of the heart off, and the heart will regenerate fully. Multiple organs. You can imagine that those are kind of hard experiments because you have to do the surgery, not kill the animal, and then it has to regenerate. So
I was getting the point that there could be some animals, then they could live forever, they won't die.
So could this make them live forever? No, because there are other reasons why they die. Not because they're in the lab, but because... but they also develop diseases. So they're relatively short-lived animals. Yes?
To what extent does the human liver regenerate?
Yeah, the human liver is an amazing organ. It can regenerate, up to 25% of it can be removed and it will regenerate. Yeah, it has an enormous regenerative capacity. And you know, who was that? Prometheus, who gave the fire to mankind, remember? And in the Greek legends, he gets his liver pecked out every night by the eagle. Sherry knows this.
I'm a liver
surgeon. Okay. and then it regenerates. So they knew something way back when. Yes? How many times can...
Yeah, if like the leg of the salamander, can you do it endlessly? Or?
Yes, you can do it as many times as you would like. Yes. It will regenerate continuously.
So if you donate once, you can possibly donate again?
Yes. Yes.
So it regenerates if you remove part of it. What if you just damaged it?
Yeah, that's a good question. So what if you damage it and form a scar? You know, I don't know too many experiments where they just damage, like a crush or something like that. But I believe then the regenerative capacity would be reduced because you have to form that blastema in order to have regeneration. The blastema is key because you can transplant the blastema to another part of their body and it will give rise to the extra limb as well.
I'm actually specifically thinking of if you, like, I don't know, had too much to drink over your lifetime. Because it often develop what cirrhosis, right, which is scarring of the liver.
That's correct.
So
Why
why
doesn't-
are you not regenerating rather than what?
So humans don't have this regenerative capacity, even in organs like the liver, because in the case of excessive drinking, you're damaging the entire liver. So it's not like you're excising a part of a healthy, so diseased tissues don't regenerate. Yeah.
Also for cirrhosis, you cause scar tissue within the liver. It's called fibrosis. And you actually can see scar tissue, and within those you'll see some regenerative nodules, but eventually this framework of scar tissue scars so much you cannot grow the liver anymore. And actually in the end stage of cirrhosis, you get an incredibly small shrunken liver, and it's just very, very scarred. It's a fibrotic liver. So you replace a lot of the liver with scar tissue from repetitive damage. It's not just drinking. Some of the hepatitis virus can do the same thing if you have chronic active hepatitis and other toxins, but you will regenerate for quite a while, but if you keep the damage up, you'll eventually lose that ability. But I don't know why that happens, but probably because we have liver stem cells.
Yes, we do have liver stem cells. Thank you. Okay, so the basic goal of regenerative medicine, because that's what I'm going to shift to now, is to somehow figure out how to reinduce the molecular program that made the tissue in the first place and now reactivate that in the disease state. And I have to caution, there's one point of caution here, and that is that we don't know much about regenerating tissues that are diseased, like Sherry had talked about the liver. That's a big problem, and I'll tell you why in a few minutes, why that's such a difficulty. What we're concentrating on for the short term is how do we create new tissues that have been acutely injured, say a spinal cord injury, and before you have a large glial scar and the patient has lost muscle mass, et cetera, because of being paralyzed, how do we regenerate that? That's a much harder question than how do we treat the acute injury, okay? And so I'm going to emphasize this concept that if we understand something about how tissues develop, then basically we're studying tissue regeneration. All right, and so now I'm going to get to the part of stem cells and cloning and some of the real-life applications. If anybody wants to stretch or anything, do you want to take a break? It's 8 o'clock. Keep on going. Is there anybody? Okay, go for it. Okay, excellent. Just like the students. Ha ha. Okay. You're so much easier to talk to. There you're working against Facebook, you know, because they're all Facebooking. Okay. Okay, so now the nuts and bolts of regenerative medicine, it begins with the story of cloning. And so I want to take a step back and talk about cloning. And for some of you, you might remember cloning in 1973. Now, can you hear that?
I have a little beggar right here. And what you want, basically, is a whole entire person connected to that nose, right? Otherwise, you get your money back. Now, did you want me to leave room for mustache? Because there's a nice area in there which it's clever. She's clever. What the doctor is doing here, we're placing down the garments because we're going to make an attempt to clone the patient directly into his suit. And that way, you know, he'll be completely dressed at the end of the operation. It's a first in cloning, and then we could all just get the hell out of here. We don't have to hang around the cloning room while he suits up.
I think it's time to check the cell structure.
Yes, that's checking the cell structure. Checking the cell structure.
Ah, checking the cell structure.
Checking the cell structure. The structure of the cell is to be checked.
I love that. Of course, we do have to check the cell structure to understand how this goes. But I was not even born then. No. I'm just brilliant. All right, so basically the idea of cloning is what Woody Allen was talking about with that nose, is that you make a genetically identical copy of either a cell or an organ. Now, way back when this was first done, this was a remarkable feat. Nowadays, people talk about cloning like it's nothing, but I want to try to give you that flavor of just how impressive it was when cloning was first achieved. And then try to emphasize, well, what did we learn from all of that way back when? So first of all, can you believe that the first organ to be cloned was a carrot? 1958, an entire carrot was generated from a single cell that was taken from the root of a carrot. Now that shows you the ability of a stem cell to give rise to an entire, in this case, carrot or organ, you can think about it. But in humans, I'm sorry, not in humans, but in mammals, this ability had to wait a number of years. The first animal to be cloned was done by John Gurdon, and he used a frog. But I'm going to give you an example more recently using a mammalian cell. Now, here's the point. First, you can take a differentiated cell, it doesn't matter from what tissue, and if you take that fully differentiated cell and you take one of the cells and you remove it from the group of the other cells, and then you introduce it into another group of cells that are different, in this case we'll say bone cells, that differentiated cell will not integrate, right? That seems almost obvious to us now. But back then it was an important control that the cell, if you put the cell in there, it wouldn't integrate. But if you took the nucleus out of that fully differentiated cell, then you could get it to integrate. And here's an example now of a pipette that's holding a cell, and the nucleus is going to be injected into it. You see the nucleus is sitting in that pipette. This is unbelievably challenging initially. Now it seems like it's much easier to do, with experience. So here's the experiment. You take a fully differentiated cell nucleus, so that means you have to take the cell, you have to separate the nucleus out of that cell, then you have to take another cell, and you hold on to its nucleus, and then you introduce it into the first cell. Does that make sense so far? So you've just transferred the nucleus of a fully differentiated cell into another cell. And that is sufficient to turn that differentiated cell into the kind of cell that the nucleus came from. And of course, it integrates fully into the tissue. So that, okay, now I went backwards. Excuse me, now I have to go through the animation again. Maybe it's a good review. So that ability means something very, very important. What it means is the nucleus of a cell contains all the information in the form of DNA to give rise to a complete new tissue, organ. We know one organism in particular, Dolly the sheep. Remember when that was achieved? So the nucleus of one cell contains all the information that's required to make that cell differentiate. And we used to think that it only went one direction. Remember that you take it from a embryonic cell and you introduce it into another embryonic cell, and then you get a fully formed Dolly the sheep. Now we know that we can do this with nuclei from multiple kinds of cells. So what is it that's in the nucleus that makes it so able to do this remarkable feat? Well, I want you to listen to John Gurdon.
I want to talk about mechanisms of nuclear reprogramming.
2006.
Now, the reason we want to know about this is that in the far distant future, I'm sure long after my time, it should, in principle, be possible to take a skin cell and to overexpress in that cell those genes which are needed to reprogram that cell back to an embryonic condition without even having to go through an egg. That's the long-term aim. But in order to achieve that, we have to know what are the mechanisms by which these nuclei are reprogrammed once you take them out and put them into an egg.
He said in the future, what did he say? Yeah, it was 2006. 2007, the first induced pluripotent cell was created. It's phenomenal. This set of experiments was done by a number of labs now, and what the researchers showed is that they could take, and now we know you can take a skin cell, a fully differentiated skin cell, and you can introduce these genes into the nucleus of that cell and turn that differentiated cell back into an embryonic stem cell-like cell. 4 genes. We used to think the nucleus is going to be so complex. There's no way. The gene regulatory networks, 4 genes. Now, the way they did that experiment first was they introduced those genes by putting them in a virus and then putting the virus into the nucleus. And you'd say, no, that's not going to work because you can't introduce these viruses. And they're right, because in their experiments with mice, this gene, c-MYC, is called a proto-oncogene. That means an oncogene, meaning it's cancer-causing. And sure enough, 20% of the mice from this experiment developed cancer. That was in 2007. In the last 2 years, we've now found that you can use a host of family members from these 4 gene families, and they can remove c-MYC, get it out of there, because that's a bad one, put in its place a gene called NANOG, and they can achieve the same effect. We now know, just recently published, that you don't even need to use a virus to introduce these genes into the cell. You can actually use the proteins. That means there's no viral particles that you'd have to put into a cell and then put that cell into a human. I have to tell you something about, scientists, they have a sense of humor. It's not so well developed, but anyway. This is Tir Nanog. That is Irish, I think it's Irish myth, and it's called the land of eternal youth. And that's where the name Nanog came from. It's a little humor. It's like Sonic the Hedgehog. Yeah. Okay, so this ability to reprogram an adult cell to become like an embryonic cell, do you realize what that means? That means you don't have to go searching for those 1 in a million stem cells. You could take any cell, presumably, and reprogram it to be an embryonic cell. It could be one of your cells, so now you don't have to worry about immune rejection. Now you might be saying to yourself, yeah, but where are we going to get all those cells? Where would we be able to move these differentiated cells back to a stem cell? Well, I am here to tell you that there are plenty of places where you can get adult stem cells. Or adult cells, yes. Fat. So you might wonder, why can't we just take these stem cells, there's a few of them, why can't we just inject them into the damaged tissues? And I think that Sherry introduced the concept of why you can't put it into a damaged liver that's fibrotic. Why can't you put it into a brain after a stroke, or a heart after a heart attack, or into the pancreas to make beta cells that produce insulin? Why can't we just inject them? And it's complicated, but I'll tell you right now that in a nutshell, it's because diseased tissues probably aren't producing the right cocktail of growth factors that will be required to keep these cells alive. So that's why we concentrate now on acute injuries, because we presume that acutely injured tissues still can produce these cocktail of growth factors that these stem cells will need. And I'll tell you about one of those in a few minutes. We also know that if cells see the wrong signals, especially if it's a stem cell, that they can go on to do very terrible things. Certainly cancer. This is one example. This is called a teratoma. This is a tumor. It's a benign tumor, luckily, but it has bone, cartilage, hair, teeth found within this teratoma. So all of those tissues, I know it's totally disgusting, which is why I like it so much. All of those tissues are able to form. So we know that cells can do this. It's just you want to make sure that they do it in the right place at the right time. Did you have a question?
Yeah, I've heard that the veterinary research in stem cells is actually progressing in some areas more rapidly than humans.
Yes. Right, so he asked the question about veterinary medicine seems to be taking advantage of stem cells in treatment nowadays, and it's absolutely true. NBC had a story not too long ago on the introduction of stem cells from a horse back into the horse to try to treat a ligament or a tendinous injury. And you might say, well, why do they get this and we don't? Because we still believe that you do it first in large animals before you do it in humans. And in this case, what they're doing is they're isolating cells. They usually suck out either blood or fat from the animal. Then they expand those cells in culture, and then they reintroduce them. Supposedly, they're isolating the stem cells. So there's a couple questions. One, how efficient is the isolation of the stem cells from these adult tissues? Not so easy. Two, when you culture cells outside of the body, you can imagine the kind of regulatory hurdles that you encounter if you're going to introduce those cells back into an animal. With a horse, maybe not so many issues as with a human. And the third is they're introducing them into an area that's been damaged. So is it a degenerative condition? Is it an acute injury? They're also just injecting them. What's the chance that the cell that you inject stays put? These are the kinds of things that we really have to know before we can do it in humans. Nonetheless, they're doing it in large animals. Yeah?
If adult differentiated stem cells are as good as any other stem cell, have we just put an end to the embryonic stem cell source issue?
Right. So the question is, if these skin cells can be reprogrammed into embryonic stem cells, can we just do away with the controversy, forget all that? To some degree. So we know that the difference between embryonic cells and adult stem cells is different in multipotency versus pluripotent, things like this. There are qualitative and quantitative differences in these cells. But the biggest hurdle I think we face in using reprogrammed cells to treat a person is that we have to reprogram those cells in a way that doesn't make the cell becomes a stem cell, but it has to be a stem cell that's under control. It can't have lost its ability to regulate differentiation. In other words, those cells, if you introduce them back into a human, you have to make sure that the cancer risk is very, very, very low, right? Because a stem cell that no longer listens to the signals that say differentiate, that is a cancer stem cell. So we have to know how to do that. And we're still working, but wait a month. There are qualitative differences. Yeah, I would have said in my lifetime, but now no. Okay, so the big question is, if we knew the cocktail of growth factors that maintain stem cells, and presumably we could add that cocktail to cells, and we could theoretically expand what would be a limited number of cells, stem cells, into many more stem cells. In this case, we're not introducing the cells themselves, we're introducing the growth factor. And I'm going to tell you about an example of that in the last few minutes now. Well, maybe not last few minutes. You're not going to get out early. Too bad for whoever was thinking that. Okay, so I want to start first with a story that isn't at Stanford. And in fact, it was from quite a long time ago. This was a research report that was published in 1948 in Veterinary Medicine. And it was done in Ithaca, New York, upstate New York. And this is the name of it, a preliminary report on the propagation of avian pneumoencephalitis virus Newcastle disease in vitro. I know you're thinking, that's a real page turner. But these scientists were very interested in figuring out what caused Newcastle disease. Now, they were at the vet college associated with Cornell, and they were studying this disease that affected the poultry. It turned out it affected all avians, so ducks as well as chickens, a number of other kinds of birds. But most importantly for the poultry industry in upstate New York was that this disease decimated the poultry. And these researchers were trying to figure out what happened in this Newcastle disease. So it's 1945, and they're working hard to figure this out. And they were very lucky. They were very diligent. They figured out how to grow, it turned out to be a virus, how to grow the virus that causes Newcastle disease in these avian cells. They worked years on this. isolating the cells from the birds, plating them out, getting them to grow. This is very hard. This is just a differentiated cells. And then infecting those cells with a virus that caused this terrible disease. And in doing these rigorous experiments, they were able to identify this virus called a paramyxovirus that caused this disease that decimated the chicken poultry farms in Ithaca, New York. And you're probably thinking now, there they are. And they're so happy to have published in veterinary medicine, and you've never heard of them. Around that same time was a disease that was the scourge of the nation, and that was polio. Most of the victims were children, and in 1952, only a couple of years after that paper was published, polio killed more children than any other communicable disease. There you see the numbers. The ones who survived oftentimes had to face a life in an iron lung. The lucky ones could walk with crutches. The public reaction was that this was a plague. I only know the stories I've heard about just how terrible and scared people were about this terrible, terrible virus. And now here's a man whose name you probably do recognize, Jonas Salk. He was working very hard on what caused polio. He joined the University of Pittsburgh in 1947 when he was in medical school, and he had to beg for a lab, so there's always a shortage of lab space. And he was working very hard on this problem. And you can imagine, this was the HIV of the times, right? Only the victims were children. It was a horrible disease. And his biggest hurdle was he was unable to study the virus because he could not grow it in cells. And without the ability to grow the virus in cells, that meant he could never generate sufficient amounts of the virus to really analyze it in detail. And so he was stuck until he saw this. He saw the paper from the two people up in Ithaca studying those chickens that were dying, and he recognized that avian cells might be more susceptible to infection with that virus, that polio virus, than were the cells that he was using. He used avian cells to infect them with polio virus, and in doing so, he began to develop the first polio vaccine. In 1952, he tried the first vaccine on his wife and child. And in 1955, 3 years, people were getting treated with that polio vaccine. 97% of reduction in the incidence of polio. What I am emphasizing here is something that I hold very dear to my heart, and that is that we stand on the shoulders of giants. That's from the Google website, you know, when you look on the Scholar. His ability to generate a vaccine depended on somebody doing research on chickens. And that's one of the most important lessons that I want to leave you with. And I'm going to now introduce this video. You need to turn up the volume.
The money that we spend, the public dollars that we already have and are spending. This is a matter of reprioritizing. We've got a $3 trillion budget in this country, and Congress spends some $18 billion on earmarks for their political pet projects, and that right there is more than the shortfall to fully fund IDEA. Where does a lot of that earmark money end up anyway? You guys have heard some of the examples of where those dollars go. You've heard about the bridges, and you've heard about some of these pet projects that really don't make a whole lot of sense. And sometimes these dollars, they go to projects having little or nothing to do with the public good. Things like fruit fly research in Paris, France. I kid you not.
So why don't we talk about fruit flies for just a few minutes here. I want to introduce you to a faculty member here at Stanford. His name is Roel Nusse. Roel hails from Amsterdam, and he works on stem cells. But that's now, and I want to tell you where he came from. We're building a lot of bricks and mortar here at Stanford, but in those buildings are these individuals that make this possible. So there he is, a number of years ago. I'm sure he'd kill me if he knew I had that picture. He doesn't look all that much different. And Roel came from the Netherlands. He visited the US back in the 70s. He took a Greyhound bus trip around the US, and he ended up in San Francisco. And he ended up in the lab of Harold Varmus, shown here on the right-hand side. These two guys were working, Harold Varmus and Mike Bishop, at UCSF on how viruses worked. And in the, let's see, I guess it was the 1980s, they won a Nobel Prize for their discovery on the origin of retroviral oncogenes. I won't go into what that is exactly, but Harold Varmus wrote a letter to Roel after he visited the lab, and here's the letter. And in this letter, he talks to Roel about the kinds of experiments that maybe he should do when he comes to the lab. And this letter is on, actually you can Google it and find it. But there's 2 points that I want to draw your attention to. The first is that he says you can work on anything, basically. Even though your fellowship says to work on a certain thing, you can work on anything here. And in the second page of this letter, he says down in that bottom right-hand corner, P.S., I have one project in particular that I think will be interesting for you to work on. That P.S. became Roel's project as a postdoc. Roel identified a gene in fruit flies, which is called wingless. Wingless gets its name, like most fruit fly genes, because of the way the embryos look when that gene is removed. So you see on the bottom that that fly has only one set of wings instead of two, and so they gave it the name wingless. He worked on this gene for many, many years, and he found out that there is a vertebrate, mammalian, homologue of wingless, and we call it Wnt. Now, it turns out here you see an example of how many Wnt genes we have in mammals, enormous number of them, 19 Wnt genes, countless receptors, countless parts of the pathway, which Roel played a big part in identifying and discovering. Now we have these kind of diagrams that show you how cells interpret a Wnt signal, what they do in response to it. It's quite complicated. I just want to show you today, I did a search. I used the criteria here, Wnt and cancer. 170 pages worth of papers on Wnt plus cancer. That's today. Then I said Wnt and stem cells. 77 pages worth. Wnt and regeneration. Let's see, how many pages was that? 19. That's today. He started off working on a fruit fly gene, and today we have hundreds of papers having to do with Wnt and cancer, Wnt and stem cells, Wnt and regeneration. I'm going to tell you one story about Wnt, and that has to do with the development and regeneration of cartilage and bone. I'm going to just focus on bone for the sake of being short. What we know now is humans who have mutations that cause too much Wnt signaling, that is, too much Wnt is either being produced or cells sense that there's more than there is, they develop a kind of condition called Van Boehem disease. And what I hope you can appreciate here is that the skeleton of a person who has too much Wnt signaling is there's too much bone. We also know that in diseases where you have bone loss, this is multiple myeloma, that bone loss is associated with too little Wnt signaling. Now, I'm being pretty brief here, but basically we now have tools in which we can assess how much Wnt does a cell see? How long does it see it? And those tools are shown here, where embryos or cells are engineered in such a way that when they see a Wnt signal, they turn blue. That means in a tissue section, you can stain it with this special method, and you can see where did the Wnt signal act on cells. And here, within bone, this is a section through bone, and that little dotted line indicates the periosteum, that's the tissue that surrounds all bone. We can see the blue cells. So we know that when those cells that are in the outer layer of the bone, if they see too much Wnt signaling, then they make a lot of osteoblasts. They make the bone-forming cells, and you get that Van Boehem disease. We know that if Wnt signaling is blocked, then bones don't heal. We also know you have bone loss. So, like the principle of Goldilocks, you have to have just the right amount. And with rule, we did some experiments where we found that Wnt signaling is activated, that blue indicates activated or turned on when the skeleton is injured. So in an intact bone, you have a little Wnt signaling, but when you injure a bone, the Wnt goes way up. So what's the purpose of that, we wondered? And we've done a whole series of experiments. I can now tell you that if you block that upregulation, then you block bone healing. So we know that Wnt signals are absolutely essential for bone repair. So now this is the Goldilocks principle, right? You know you have to have a little bit, not too much, not too little, otherwise you get these diseases. And in 2003, after nearly 15 years of trying, they purified for the first time the Wnt protein itself. It took 15 years. This person, the rule never gave up on this. And in doing that, he opened the door to be able to ask a simple question. What if we take that protein and we put it into an injury, a bone injury that doesn't heal? And in my own lab, we figured out a mechanism for packaging the protein. I won't go into that now. And then we introduced that packaged Wnt protein into skeletal injuries. And here you see that in the side on the right, which has gotten the Wnt, you see there are more blue cells. That's important because that tells us that what we introduced turned up the Wnt pathway more than normal. And what we found is that bones that were injured healed much, much faster than bones that were left alone. We know that they make 350% more bone than injuries that are just left go. Now you might say, why do you care about healing a mouse bone? I don't. What I care about is healing a human skeletal injuries that are slow to repair. Most of us, you can say we're lucky. When we have an injury, we regenerate bone fairly well if we're healthy. But as we get older, as we have a disease, if we're immunocompromised, then that ability is really repressed. This may be a mechanism whereby we can stimulate bone formation, and we now know why it works. Because it turns out that the stem cells within an injury site respond to the Wnt by proliferating. That's the key feature, and then those cells differentiate. Now, the important thing here is this balance. We know everything's a balance, but we know that cells tend to make more bone faster when they see a Wnt signal. And so this cell in the center is the stem cell that seems to be pushed towards a bone-forming fate. Now this is the state of art of cartilage regeneration. That's it, right there. So there's a lot of room to improve here and see if we can regenerate cartilage tissues through a mechanism. But I want to leave you with a very important point here. I told you about one tissue, bone, and Wnt in one tissue. But here's how far that fruit fly research that has no value to humans, right? How far it's gone. Here's in nature, Wnt-dependent hair follicle regeneration, for those of you who are a little bald on top. It also seems to accelerate skin wound healing. Here's another one. It mediates the proliferation of neural stem cells after stroke. Another paper, the role for Wnt signaling in the regeneration and self-renewal of hematopoietic stem cells. So these are in the top-rated journals in the world, and you can see that the regenerative potential of this fruit fly gene is tremendous. Now, I want to leave you with this recent paper that says this Wnt pathway is required for patterning in planaria. Does anybody know what a planaria is? A flatworm. Now, you're probably saying to yourself, why should I care about a planaria? And these animals, you can cut them in half. Do you see the eyes here, the little dots up there in the corner? Those are the eyes. If you cut this worm in half, it'll regenerate a tail. But if you tweak the wind pathway, it'll regenerate 2 heads. Pretty cool. And if you cut off the head and get rid of that and keep the tail, it'll regenerate 2 tails. Wind signaling regulates that all the way down to flatworms. And you might say, why do I care about a flatworm? Oh, but I hope you don't. Because nobody knows where this is going to lead. So I think in conclusion, we are living in the most exciting time in stem cell research. I can't imagine a better time to be involved in science. I think the hope of regenerative medicine is firmly established in basic science research. It needs that foundation. So people who think about funding research for all kinds of animals, whether it be fruit flies, flatworms, or chickens, it does make a difference for human biology. And I want to emphasize that it's not necessarily a straight path from the bench to the bedside, and being willing to go along for the ride is one of the most important attributes of a scientist who's in this field, because you have to believe in the long term that it is an enormous potential we hold. And this is the last quote from Governor Schwarzenegger, so you can see on both sides of the political debate that he thinks that he's a great believer. It will improve not only the human condition, but our economy as well. How good is that? Thank you very much.
I think you can see why we asked Dr. Helms to speak tonight. And thank you for telling us why planaria are so important. And thank you to our bone marrow expert in the audience there.
Right there.
We have some time for questions, so please ask away.
Yes.
You spoke most of the stem cell rebuilding organs happening within the body. I've seen you work on organs being grown outside of the body, also either for ear being grown in the back of the mouth. So is that the same phenomenon? How is that different? Is that any farther along than, I lose my ear, that as opposed to growing more?
Right. Right. So the idea of can we regenerate tissues outside of the body and then bring them back in? And he reminded me of the mouse with the ear growing on its back. So first I have to tell you what that mouse growing the ear on its back. So that is actually not in the ear. So what that is a piece of cartilage that was tucked under the skin of a mouse, and the skin then formed around the cartilage. Now that mouse you remember it didn't have any fur, so you could really see the ear. That's because the cartilage came from a human, okay? And so the mouse had to have a suppressed immune system. So that wasn't an organ. It didn't have any hearing capabilities. It was merely the architecture, the outer architecture of the ear. And now you have to realize that while the cartilage was human, the skin was mouse. So there's no way to introduce that back into a human. So that was just a demonstration, I think, that is it possible to grow tissues that have sort of shapes to them that look appropriate, but not functionality. So there's no hair follicle regeneration, inner ear hair follicles. There's no functional restoration of hearing, nothing like that. All right, so that's it. Now, can we grow an organ in a dish? The answer is no right now. And that may be because of what we need in order to grow an organ. Nothing will grow without a blood supply. And we simply cannot create that in a dish. So try as we might, most of our experiments have to be done in an animal. And that's another reason why I think anybody who does research has to think about, what's the balance between I'm using a mouse, this mouse will die. What's the advantage of it? Because that animal will die for the hope of doing something that will benefit mankind. So without a blood supply, we can't grow an organ in a dish. There are a lot of other limitations, like we need a lot of different kinds of cells. And there are also the contribution of things like They get carried along with the blood, not only the blood itself, but also all of the growth factors. We can't recapitulate that in a dish. Yeah. Yeah?
A couple years ago, I heard of someone doing research into pig hearts. They were taking the pig hearts and using some type of detergent to wash out the cells and leaving the remaining structure, and then somehow repopulating that with human cells.
Mm-hmm.
Do you know anything about that?
Yes. So he was reminding me of the pig heart, where they isolated pig heart, then they remove as many of the cells as possible, leaving sort of the structure of the heart, and then they wanted to repopulate that scaffold with human cells and then introduce it back. These are called xenografts. And so they attempted them in a number of different kinds of tissues, from valves to an entire heart. And the basic limitation of that, there's a big advantage. The structure is there, right? And that's hard to rebuild, right? I mean, mechanically, the man-made heart is, a far cry from the functionality of a pig heart. The real limitation is, though, that again, one has to think about the immune rejection part, right? So if you're replacing an entire heart, yeah, that might be the strategy to use. But what we hope is that the diseases that lead to needing a complete heart transplant may be circumvented by treating illnesses very early on. Like, you remember the picture of the young man with the amputated leg and the artificial leg. Somebody might say, well, you can't regenerate a whole leg. But what you have to realize is the amputation, just like this heart transplant, is not the result of a single damage to the leg. It's an infection at the site of injury. The infection can't be controlled, so they have to cut out tissue, which means that the wound doesn't heal, leading to further infection, another surgery, et cetera, and then you get amputation. Same way with some of the heart diseases that require transplantation. Can we intervene early enough to prevent the need for that? But yes?
How is the wind delivered? Is that by injection?
Right, so how is the wind delivered? So we've now treated bone, cartilage, skin, muscle, a heart muscle, brain, retina, and cornea with this approach. And in some cases, it's topically applied if it's the skin. In other cases, it's injected through a catheter if it's the heart. In bone, we can put it on things like collagen sponges that are put into injury sites. That's what's currently used. Not because the collagen sponge is anything special, but just because it's sort of a delivery vehicle. We have to figure out ways to optimally package it, and for each application, and we're still learning that. So in some cases, it's injected like following stroke into the injured area, the mouse brain. In other cases, topically or in some other solid support. Yes?
I thought I'd give you the opportunity to do like John Gruden did in that video that you showed. When do you think that we might have humans regrowing feet or legs like you saw that movie?
What were the words he used?
Not in his lifetime.
Not in his lifetime. Maybe not in my lifetime, but in the lifetime of children that are alive today. Yeah. I think, I can't believe that I could actually say that, but I really do. I see the progress is at a speed that's almost unbelievable. I had slides from a lecture that I gave 6 months ago on the regeneration, with the different cell types. That's already changed. That was 6 months. I mean, these are huge, huge advances. So I never believed a differentiated cell could be turned back in time. So I don't know. I think the limitation is not like it used to be.
We have questions from this half.
Ah, I'm sorry.
It's hard. The lights hit you here. That's why nobody wants to look this way.
Yes.
I'm just wondering why somebody hasn't taken Sarah Palin aside and explained to her. And that sounds funny, but that really is a serious question. I mean, we sort of, it doesn't take that long. What's the obstacle that keeps
opens and I can walk through. Well, I think there's two things to be kind. There's two things. First, you have to be willing to listen. And I think the fact is a lot of people do not want to listen. So, and then you have to, well, besides being willing to listen, you have to be smart enough to understand. Yes.
Is there any possible rejection of the when it's added or is it similar to what's already in the body? And then also in an instance, you remove the nucleus of the cell and reposition. What happens to the cell, the residual cell, after the nucleus is no longer present in it?
Right.
Does that regrow or does the nucleus develop?
Yeah. So all right, so two questions. One is, if you take the nucleus out of a cell, does it regrow a nucleus? The answer is no. You've removed all of the information for survival in that nucleus, so that cell is gone. All right, and then you asked a question about the Wnt. About what was it now? I'm sorry.
Presumably, if you add it, is there any rejection?
Oh, is there a rejection? No, there isn't a rejection for the simple fact that you're not introducing a cell, you're introducing a protein. It'd be like injecting insulin. We don't develop immune response to proteins. So that's the big difference. Yeah, no cells. Yeah?
You mentioned the spinal cord in regeneration a couple of times, and I know there's going to be the first human trial related to acute spinal cord injury that should be starting soon. Is the spinal cord a particularly good model, or why is the spinal cord injury the first FDA-approved human trial?
Right, so why is the spinal cord going to be the first, and is it somehow a particularly good model? Well, first of all, it's a devastating injury. Second, there's no treatment. These are the kinds of conditions that make it easier to get FDA approval versus doing something that a person could live without. So there's no treatment. Third, acute injuries are going to be the first things get tried versus chronic degenerative diseases. Because of this, the acute injury site shouldn't have developed too much of a scar. There still will be a scar, but it will be a better site than, say, one where chronic disease has led to the tissues atrophying, et cetera. So it's not only good because it's so devastating, but it has no treatment, and acute injuries are a good model.
How about our last two questions? The lady with glasses, and then there's a gentleman sitting right underneath the very bright light.
Wouldn't be able to see him.
Yes.
So my question, to start at the beginning of the lecture, what's the dealio with people saving umbilical blood? It seems kind of creepy.
What's the dealio? So why do people save umbilical cord blood? Because in the cord blood are stem cells. Now, to what extent are those stem cells viable through the process of harvesting? Usually this tissue is discarded.
Well, haven't they been differentiated? I thought it was only 5 days you died.
So some of the cells, you could call them tissue-specific stem cells, right? So they aren't embryonic stem cells. They're more tissue. At that point, they're called adult stem cells, meaning anything post-embryonic, day 5. So they appear to have some regenerative capacity. And so what people are doing is banking this umbilical cord blood for their child on the chance that technology will have progressed to a point that should they have a disease or an injury that could be treated by stem cells, they have a bank of their own stem cells from them.
In a drawer at home or something?
Not in a drawer at home, no. There are...
You have to rent space for them.
One gnarly drawer. Yeah, it's exactly right.
Yeah.
You have to rent space for them.
It's
so there are
expensive.
companies. Yeah.
And the gentleman in the blue shirt.
I was wondering why we had tissue-specific stem cells versus universal stem cells everywhere. What's the advantage of that?
Right. What's the advantage of universal stem cells? Well, why do we have tissue-specific stem cells? Why not universal? We have universal stem cells when we're 5 days old in that inner cell mass. That could be considered the time when we have stem cells that are universal. Why do we have tissue-specific stem cells? It's a good question. I don't know why. but it does appear that every tissue harbors stem cells. So tissues that we once said there's no way that they have any regenerative capacity, the brain, the heart, it appears that in fact they do. And in some cases, those stem cells in the brain and heart and other tissues like photoreceptors of the eye, they can be reinduced to somehow give rise to more photoreceptors, say, that have been lost because of an acute injury. So why we have them, I don't know. Thank God we do. And they seem to be the key to maybe how particular tissues might be able to heal.
So what I'd like to do is, you're going to see Dr. Helms next week because neither myself or Dr. Pizzo can be here. So she's going to be introducing our next speaker, who's going to be speaking on the human nervous system. I would highly encourage you to read the chapters assigned for next week so you can really follow along. And again, I'd like to thank Dr. Helms for such a fabulous presentation.
For more, please visit us at stanford.edu.